Okay, welcome to Life Science Cafe. My name is Natasha de la Rosa. I am a fourth year PhD student in the Neuroscience and Behavior program at the University of Massachusetts Amherst. Oh, give me one. Hi, Emma. Okay, so I will be your MC for today. So the Life Science Cafe was established in 2011 and is organized by graduate student researchers at the University of Massachusetts Amherst. Each month, we invite different experts to discuss their research. Typically, we host these events in the Amherst community, but we are happy to continue this effort virtually in this interesting time. We would like to thank our funders, the Organismic and Evolutionary Biology Program, the Graduate Student Senate, the Graduate School at UMass, the National Science Foundation, and our generous UMass Gift donors. For our next event, we will be discussing climate impacts on fish ecophysiology with Leanne Guo on October 21st. Subscribe to our mailing list to stay tuned for future cafes. And help me to introduce and welcome Emma Dauster here today, which will be talking about how neurological disorders manifest differently depending on biological sex and how we can study these differences. Emma is currently a PhD candidate at UMass Amherst.
Hi, Natasha. So nice to see you again.
Hi, Emma. Okay, so would you like to share your slides or do you want me to share them for you?
I think I can share them when the time comes. I don't want to distract people.
Sounds good. So let's start. Can you tell us a little bit more about yourself and what is motivating you to pursue a PhD in neuroscience? And that's just a PhD overall.
Okay, so my name's Emma. I'm a PhD candidate, as Natasha said, and I'm studying neuroscience and behavior because I feel like we have solved a lot of problems over the years, which is really great, really amazing. In the developed world, we have access to a lot of vaccines and treatments that used to kill people much younger in life on average. And now we're living a lot older than we used to. So as a people. So that means that in a lot of cases, our brains aren't able to keep up with how long we are living in the rest of our bodies. And that's kind of what inspired me to think, well, maybe the brain is like the next frontier of research. And we have all of these new technologies that in really the last few decades, we've increased our ability to even study the brain at all because it's behind this big skull, right? So it was hard to get to for a long time. And now we're at a really exciting time in neuroscience where we can actually study these things. But yeah, so that's why neuroscience is interesting to me. But I have been getting my PhD because I've been interested in research for a really long time. And it's the kind of thing where if you can be involved in research with absolutely no experience, it's really awesome in that way. But I've been told by a lot of my mentors that you will reach a ceiling of where you can go with your research if you don't get a PhD. So you can get a career and do all these great things, but the PhD really opens a lot of opportunities for you to advance in your career. So that's why I'm getting my PhD.
That's pretty cool and pretty relatable as well for me. So you study neuroscience, you study the brain. So what is your research on a little bit more specifically?
All right, very broadly, I study how female and male brains compare to each other. And I study that in rats. And I when I talk about females and brains, I'm going to make a disclaimer right now. Did I just say females and brains? Females and males in brains. I'm going to make a disclaimer that when in our lab, we identify females and males based on their anatomy, based on their appearance and their bodies. So males have testes and females do not. Females are about half the size of males in rats. And so that's what we're basing this definition on in our research, but there's a lot of different ways to define males and females. And there are a lot of other options outside of the binary that I'm not working with in my research, but hopefully research will progress to that in other in the future. So we're just starting by including females in the conversation in the first place. But yeah, we're trying to figure out how to make more effective treatments for neglected parts of the population that just weren't involved in research before.
I think that's amazing. And I'm so glad that there's people out there like yourself that are working on this and focusing on this. And I appreciate your disclaimer and how and defining how you are utilizing biological sex and how you're characterizing that. So that's super cool. So I'm sure you actually do a lot of research and you've done a lot of like research work and many different experiments throughout your doctoral career. So what exactly are your questions for your dissertation at the moment?
All right, so this is where I'm going to share my screen because we're getting into brain stuff that might be a little confusing. So can you see a graph that you're not supposed to see right now?
Yes, and I see Tetris, which I am so down.
Yeah, you know it. So that's when I'm hard at work. Sometimes you need a Tetris break. So I'm talking about the brain. And I'm gonna present it so that you can see a little bit better. I know there's a way to do this.
Yes, top corner. Yes, top corner to the right.
Behind our faces on my screen. That's why. Okay, so I study the brain. This is an overview of your brain, very, very simplistic, just to show that different parts of your brain are involved in regulating different kinds of behaviors. So these are different parts of your brain that do much more than the thing that I labeled it as. And these behaviors that I've labeled are also controlled by many other parts of the brain that are not in this diagram, but just as an overview, there's a lot of different behaviors that your brain can control, and there are different parts of your brain that are that have been linked to these kinds of behaviors. So I'm going to focus on this little part of the brain down here. It's kind of where your brain, it's close to where it meets your spine. So it's called the locus coeruleus. And the reason that I focus on this little part of the brain is because it's the main source of a chemical in your brain called noradrenaline. It's also called norepinephrine. So if I mix up one or the other, just know it's the same thing. For some reason, it gets two names because it's that important. So it's the main source of norepinephrine for the entire brain. So it sends this chemical all over the brain. And these cells are also really heavily involved in arousal behavior. So this is everything from when you go to sleep and when you wake up and when you pay attention and all sorts of stuff like that. So that's what this part of the brain is really involved in. And that's like one of my big focuses is trying to figure out, okay, how does this part of the brain look in female and male rats? And what I study specifically for this talk, I'm trying to focus in so it doesn't take too long, is how this part of the brain communicates with the front of your brain, right behind where your forehead is. There's a part of the brain called the prefrontal cortex, and that's really heavily involved in regulating how you pay attention to stuff or how you don't pay attention to stuff. So that is a big link from the locus coeruleus to the prefrontal cortex. It sends norepinephrine to the front of your brain, and I study how that relationship happens and how that allows individuals to pay attention to stuff or not. So if you think about attention, one of the things that a lot of people think about is attention deficit hyperactivity disorder, which is something that's diagnosed in humans. It's not diagnosed in rats because they're rats. And we find that when you look at diagnosis rates of ADHD, there's way more males diagnosed than females. And that is likely due to a lot of different reasons. So that's one of the big reasons that I study rats instead of humans, is to try to separate the societal impacts versus what's biological and anatomical and chemical, and are there of these various explanations, what are some reasons that we're seeing these sex differences in diagnosis? So, like I said, I study the rat. We've got very similar parts of the brain in the human brain and the rat brain. They both have LCs, the locus coeruleus, and they both have norepinephrine that gets sent all over the brain. So these arrows are all where norepinephrine goes in the brain, including the prefrontal cortex. So if you study the rat, you learn a lot about what could be going on in the human brain as well. And we have found a lot of biological differences and anatomical differences in the locus coeruleus when you look at females and males. So this is looking at the rat now. We've got way more cells in the female locus coeruleus than the male locus coeruleus. And those cells have way more, they're just set up to take in more information from the cells around them. So if the cells near them are releasing a bunch of chemicals, the female locus coeruleus can grab those chemicals and take that information from around them. So we don't know what other sex differences there are in this part of the brain, and that's part of what I wanted to answer in my PhD. So I'll stop there for now so that we can not spend all day on it.
Yeah, that was very interesting and a lot of information for sure. So, okay, so there is a bias of like diagnosing a lot more males and humans with ADHD. And so, but there seems to be some biological differences in these. And so why do you think these differences haven't been studied before, even though it seems pretty apparent?
Well, so it's apparent after you start finding that it's apparent, right? And not everyone's going to ask the question if there's a difference between a male and female brain, because it just doesn't occur to everybody to ask that kind of question. Historically, research has been really male dominated. So if you have a bunch of men in a room, they're going to ask questions that they generate in their brains based on their lives and their experiences, and there's nothing wrong with that because everybody has their own biases that they bring to the table. But when you increase the diversity of the researchers who are asking questions, then you get to fill in those gaps that people were not thinking of because it didn't really occur to them based on their life experiences. So we have a lot more female researchers who are coming to the table and saying, well, maybe there are some differences, and let's just even look at the female brain, because it might surprise some people to find that research has pretty much exclusively been done in males in a lot of aspects of research. It's getting a lot better these days. In 2019, I have another slide I can show actually. There was a really wonderful study. One of the authors on this paper, it actually works down the street in Northampton, and Annalise Berry, she is amazing. But even if she weren't an author on this paper at Smith College, I still think it'd be an excellent paper because it shows a survey of all a bunch of different biological research disciplines, and it breaks it down over the last 10 years. She did one in the previous years as well. So this is a follow-up study to be more recent from 2019. And it breaks it down male, female, both sexes, and unspecified. And based on a history of biology research, unspecified basically means male only. And if you add the blue and the gray, it's pretty much 50% of the biological research that is being done today. So we're a lot better than zero, but still there's a lot of progress that needs to be done. So weren't asking the question. We didn't know how male and female brains compared before. So that's why we didn't know, because we weren't asking.
That makes sense. And I like that there's data like this out there, like gathering this information, like forms of survey and this, you know, taking a brief history of what our research has produced. It's pretty nice. So we'll take a small break right now to have a Q&A time period. And this is the time for the audience to ask any questions that you might have for Emma. So yes. How was your day today? Where are you at right now?
I'm in Denver, Colorado right now, which is crazy. So when I said right down the street, I was pretending and envisioning I was back in Amherst, but I'm living the remote life right now. I'm so excited to be back involved with Life Science Cafe for the happy 10 years. It's very exciting, and it's so wonderful to see you guys again.
Yes, we are excited that you are our first speaker as like previous member of Science Cafe, and you know, making it so great and what it is today.
Thanks for inviting me.
So there's one question. How did you start working with rats?
That's an excellent question. So when I first started doing research, I was studying yeast cells, like single cell, what you make bread out of. And it smelled really good. And it was a very nice, like, ease into research. And I was working at the National Institutes of Health, and I wanted to get like a t-shirt to kind of commemorate my experience. So I found one that had a rat wearing a lab coat, and I thought that was really cute. And everyone was like, "Oh, do you study rats?" And I said, "No, I could never study rats. They're so gross. Like, no, never." But then when I went off to college, I was looking into which labs I wanted to join and what kind of research they were doing that sounded interesting to me. And like I said earlier, I really thought that neuroscience was an interesting direction that I wanted to go in. It really allowed me to take what I knew about how cells work and how genetics work and see how it applies to behavior, which you can't always do that. But in neuroscience, it's a lot easier. So that's why I went, joined the lab that I did that was asking interesting questions, and they just happened to be working with rats. So I was really intimidated when I first started, like on the first day. My advisor was like, "Okay, here's a skull, take the brain out of a rat skull." And I was like, "I don't think I could do that," but I actually did, and it was no problem. And when you challenge yourself like that, you'll be surprised with what you can accomplish.
Oh yeah, definitely. So what besides anatomical differences between males and females, have you seen any, or do you know of any behavioral differences between sexes of ADHD, like rat behavior?
Yeah, so I did a lot of research on that. I have found that they behave differently. A lot of that research has to do with when you manipulate the different chemicals that they have in their brain, because what we're finding is that females and males seem to have a different baseline amount of chemicals like norepinephrine in their brain. So females just might have more circulating in their brain, and then that might be protective of certain attention deficit behaviors. So what we do a lot is a decision-making task, where we have these levers, and we shine a light, whether it's a green light or a red light. We have the rat either press the left lever or the right lever in a little behavior box to see if they can learn that task and pay attention to which light is flashing. And we see that their percent corrects in that task are different depending on which chemicals we're manipulating. And we see that sometimes males will have more inattentive trials than the females, and different behaviors like that. So it's not, we can't say that these rats have ADHD, but some of the attention related behaviors that we're seeing are different in ways that would be expected if you compared it to what we see in humans.
I see. That sounds very interesting. Thank you. One last question before we go into our next vignette. Given that ADHD is diagnosed in humans based off life history rather than any biological metric, how can we know that the rat model accurately represents ADHD?
Yeah, I think that's a really great question. And it's really important to remember when you're studying animals that, I mean, humans are animals, but when you're studying non-human organisms, that they are not human. So whatever you're learning about them is what you're learning about how a rat works. And we can do comparative studies to see, okay, well, how does a human work? And the more we see that overlaps with each other, the more we can understand, okay, well, if this happens in a rat and that other thing happens in a rat, like what I said about the number of cells in the locus coeruleus in females compared to males, that's been found in rats and in humans. And so when we see other sex differences in the locus coeruleus of a rat, we can say, okay, it might be that way in humans. And it's very possible because we've seen other similarities, but what we're seeing is what we're seeing in rats. So it's really important to keep that in mind.
That makes a lot of sense. But it sounds like it's still super helpful as a helpful model for a lot of other things. Thank you, Emma. So to go into our next vignette, we will go a little bit more into your dissertation and what it's about. So we keep throwing around ADHD, but what exactly is this? How is it diagnosed? And how is it different from ADD?
Okay, so like I said, I study rats. I'm not diagnosing anybody in my day-to-day, so don't come to me with diagnosis questions. But in humans, there's something called the Diagnostic and Statistical Manual of Mental Disorders, so the DSM for short. There's many different editions. I think we're on DSM-5 at this point. And what that diagnostic manual says is that people with ADHD show a persistent pattern of inattention and or hyperactivity impulsivity that interferes with functioning or development. And they have a bunch of different behaviors that you can look out for as if you're a professional diagnosing someone, not just anyone on the street. That's how people get diagnosed. The standard forms of ADHD, there's several different kinds. So there's hyperactive, inattentive, or there's also both. And I believe that the term ADD is an old term that used to be used to describe the inattentive version of ADHD. But these days, I think people don't use it as much anymore.
I see. That actually, I don't know. I didn't know that. That's cool. Thank you. So how common is ADHD overall in humans, I guess, obviously?
So based on diagnosis rates, which seem to be increasing as we increase awareness, but the CDC, the Center for Disease Control, says that 9.4% of children are diagnosed with ADHD today. There isn't that much data on adults, so I don't have the stats for that, but it is something that people carry into adulthood with them as well.
Yeah, no, there's definitely an increase of a lot of diagnoses, it seems like. So going a little bit back to sex differences, what else can you tell us about sex differences in ADHD and that you have found?
So I haven't been studying ADHD, but I can tell you about other sex differences that I found in rat brains. And so I've looked at behavior, which I talked a little bit about, and what I've done to manipulate these brain chemicals, which I kind of just mentioned, but didn't actually talk about how I would do that. I give my rats treatments that humans will take. So things like Ritalin, Strattera, things that manipulate the amount of chemicals in your brain that are actively available to be acting in the prefrontal cortex. And so I'll give my rats these different chemicals, and I found that males seem to be in this decision-making task that I was talking about earlier with the levers. They seem to be responding more to an increase in norepinephrine, while females don't have much response to norepinephrine alone, but they do respond more to an increase in both norepinephrine and Ritalin. And so that just comes down to which drug I'm giving them. So the drug that just looks at norepinephrine is called Strattera, or atomoxetine. And the drug that manipulates both of them, and there's the drug that I use, there's many drugs that do these things. I use Ritalin to manipulate both dopamine and norepinephrine, and it's also called methylphenidate. But you guys might be more familiar with Adderall as well. It works in a very similar fashion, but it also increases the release of the chemical. So it doesn't just make it more available, but it like actually makes more of it. It makes it more available in many different ways. So I found that they behave differently when we manipulate these chemicals in their brains. I've also looked at their brains aside from their behavior to see how the locus coeruleus sends norepinephrine to the prefrontal cortex. And what happens is, you saw the diagram that the locus coeruleus is way far away from the prefrontal cortex in the brain. So it basically, the cells in the locus coeruleus will like extend a really long arm and like drop off norepinephrine in the prefrontal cortex. So that arm is called an axon. And I just looked at the density, like how many axons are there in the prefrontal cortex in females and males that are coming from the locus coeruleus. And it looks like there's more female, it's denser projections in the female prefrontal cortex. So they might have more ability to drop off norepinephrine in the female prefrontal cortex than the male. And we're looking at the different receptors that like take that chemical in when they're in the prefrontal cortex. So like, just because you drop something off doesn't mean someone else is going to pick it up. So yeah, we're finding all sorts of sex differences. It's not just limited to the back of the brain.
Yeah, that's interesting. I was thinking here about like, it's like a highway with a lot of lanes. The more lanes you have, the more cars that can go through, and the cars have like the neurotransmitters. But I know what neurotransmitters are, but what is dopamine and noradrenaline / norepinephrine, and what are neurotransmitters?
Okay, neurotransmitters are chemicals in the brain. So they transmit signals, information. And so dopamine and norepinephrine are just examples of those neurotransmitters. I'm trying to think what else I had to say about dopamine and norepinephrine. They do a lot of similar stuff. They're actually very related chemically. So if you look at like the chemical molecule, I don't know if you guys care about this, so I won't go into too much detail, but they're very similar in a lot of ways. So it's not surprising that they are both involved in regulating things like attention.
That makes a lot of sense. And so, I guess like, they are similar, but they still behave somewhat differently, considering that they're different drugs that affect them, like the Strattera and the Ritalin. So So before we leave the theme of drugs and prescriptions, are Ritalin and Strattera like the common drugs that are prescribed to humans? Or
Yeah. I use the same stuff you guys might use. And that's another reason that we don't use Adderall in my studies, is because it's just not worth the headache of, it's like a higher, what's that rating system that it's like a higher potential for abuse. So it takes more paperwork for us to work with that drug. And since the chemicals that it affects are the same as Ritalin, we just use Ritalin.
That makes sense. That makes a lot of sense. Okay, so stepping a little bit back and more into ADHD, and I know you're not a diagnostician in any way, but how is it that somebody differentiates between being easily distracted or having ADHD?
Okay, so one of my favorite findings from my PhD is that if an individual is taking this drug, that does not automatically improve their performance. Individuals that are experiencing or exhibiting attention deficits can benefit from these drugs, but if you're not, then it might not have much of an impact on your performance. I do think that the placebo effect is a really great thing. It is a real phenomenon, and it helps a lot of people. So that's really great. But that's another wonderful thing about studying rats, is that they don't know what I'm giving them. They don't know what it's supposed to do. So that placebo effect probably isn't as strong in my rats than it would be on a person who's taking these drugs. So yeah, if you're like taking it as a cognitive enhancer, which a lot of people do, then, you know, like take that with a grain of salt, because it may or may not have the effects that you would expect, although placebo effects are great. So when it comes to people who are paying attention or not paying attention, and ADHD diagnosis and stuff like that, we know that there's a lot of social perceptions that go into that. And like I said, we're seeing an increase, especially in adult women today, because those are like an intersection of two categories of people that haven't been really paid attention to as much in the ADHD community as they are now. And yeah, so it's possible they're just being noticed more, but there are people who also will benefit from taking these drugs. And if you do benefit, maybe, and you haven't been diagnosed with ADHD, maybe you are just awaiting a diagnosis, who knows, maybe it's the placebo effect, I don't know, I don't study people, but I don't think people should take drugs that aren't prescribed to them. I'm gonna leave it at that. I do think that there, you know, whether or not you're feeling distracted, treatment is totally a personal decision. You know, not everybody wants a treatment, not everybody needs a treatment. That's totally a personal decision, and it's really important to that like there are options for different environments, different learning environments, different working environments that work for different people.
Yeah, no, that makes a lot of sense. And I was definitely gonna say something and I forgot, but I guess it goes hands in hands with like if you're only who's doing the research and what type of questions and what type of behaviors are we looking at. So but so do we really want everybody to behave the same, I guess, in terms of like attentive disorders? Like, is it not wrong? Is it, do we want everybody to be at the same attentive level?
I think like what a boring world that would be, and like a useless world if everyone acted exactly the same, right? Like you need the different people that have different strengths and weaknesses to make a successful society. So I don't think that it's the kind of thing where we want a homogenous group of people, and we shouldn't be putting that expectation, because who decides what the optimal attention is, right? So yeah, like that's why I don't use words like normal, because like what does that even mean? And it's not, I don't think that it accomplishes anything valuable in a society to think like that.
Yes, I definitely agree with that. before we leave this vignette, a lot of your research focused on creating parity across sex. And so, and how, and treatments as well. So what are some of the broader impacts of your research that we have touched lightly upon, but can you expand on them?
Yeah, so I'm hoping to make it so that we have more personalized medicine, we're getting closer to it, that people who have been largely, or groups of individuals who have been largely ignored from research historically are starting to get closer to being recognized, and that research actually applies to more people than the very small group that it has, that it does today, really. It's kind of surprising that once you get toward clinical trials, so like once you start testing treatments on humans, that's where you get even more diversity in the group that you're looking at, which it's so easy to include female rats in your studies, like it doesn't make any difference at all, but people just haven't done it as much in the past as they could. So I'm hoping to just bring light to how we can be looking at as many different groups as possible, and how that can help treat people more effectively, hopefully.
That's great. I think that was such a great argument too, like the one that you highlighted on, like, we are so concerned about, outsourcing all this variability out in the research stage, but then when we get to the clinical stage, it's like so all of a sudden you get introduced all this variability that you didn't account for, and you could have, easily accounted for some of that by just including females or some other factors. Yeah, I think that was really smart. So let's time for Q&A questions, Q&A time period. And for the first question is, do you know how these sex differences in the brain also apply to other neurodivergent conditions like autism?
Yeah, so there's a lot of stuff that this could impact. There's, for example, Alzheimer's is another thing that is diagnosed differently in males and females, and this one actually is more diagnosed in females than males. And that's the part of the brain that controls memory, is another strong place where the locus coeruleus sends norepinephrine. So the sex differences that we see in the locus coeruleus could have impacts for all sorts of different behaviors. Autism is one of those things that we're just now starting to research. That it really wasn't very commonly looked into at all historically. So there's so much that we don't know about it yet, in terms of how the brain chemicals are involved, what parts of the brain are involved, things like that. We're really in the early stages. So hopefully that research progresses, and we can learn even more about that.
Wow, it sounds very exciting in terms of like your research, and even like more research that your research could like guide you towards. Another question from the audience. Do you think that hormones may be involved broadly in the different effects to different drugs that you're seeing?
Yeah, that's a really good question. So nothing is happening in a vacuum. That part of the reason that I love neuroscience is that you can study things at whatever level you want to study them at. So there's people who look at the cellular level and they're seeing, okay, well, if you just add Ritalin to this cell or this part of the brain, then what are the impacts of these different chemicals around it? People have done those studies and they see that estrogen actually is really involved in that norepinephrine release process. I don't remember exactly how it interacts with it, but I know that there are studies that hormones are involved in this process. And what I study is systemic. So I look at just like what would happen if a person took a drug, you just consume it and it's processing through your entire body. So like norepinephrine doesn't just act in the brain, some of it acts in the heart, and you've got it's like these impacts are going to be impacting a lot of different systems. So yeah, it's important to keep that in mind. There's a lot of other factors going on.
That definitely complicates the context. We have one question into your data, and they might have missed it, but what are you seeing in terms of different treatments? How are the rats behaving differently compared to controls when you give them treatment?
So I think I already talked about how males and females are responding differently to different drugs. What I do is I give them different doses as well. So that when you give them a lot of the drug, then they have an even stronger response in my experiments. They don't have too much of a response to the lower doses. Yeah, I don't know what specifically they wanted to know though.
Yeah, no, that definitely gives us more information onto your results. And yeah, I remember you talking about this earlier, and how sometimes when you can tell if you might have ADHD as opposed to easily distracted, is how you're impacted by these treatments. And I can see how that's analogous to like how the controls and ADHD like rats might be experiencing different behaviors to these treatments. Awesome, thank you so much, Emma. So let's go on to your next vignette. Let's focus more into like what you're doing right now, and like what is the Emma from today doing at this point in her life? Like what are your plans for the future? But let's start with today.
Oh, today, I woke up and I thought, oh, I'm so lucky to be alive. It's tough out there, but I think that there's a lot of good happening. So I'm trying to do more good in the world. I'm trying to do my research and publish my results so that if you had follow-up questions, then you can read about it, hopefully in the future. It's a long process in research, as you know, Natasha. So it's taking a long time, but that's a future step. I do a lot of science publication that is not through academic journals. I do more popular science writing and communication. So that's kind of my way to make the world a better place. Hopefully, I'm trying to increase access to information and increase the diversity of people who think of themselves as scientists. So that's why I've been working. Well, one of the reasons why I've been working with a group called SciShow, which you can find on YouTube for free. Anything you want to learn about science related, they've got a lot of channels that talk about all sorts of different stuff. But yeah, I'm just trying to increase science communication, science awareness.
That sounds awesome, Emma. Like, especially because, science should be for the people, and like, people that funds it, which is the citizenry. And also like, if y'all don't know about SciShow, like this is amazing. Like I haven't seen their science shows yet, but their history channels are also super cool. Like Emma is onto some famous steps over here.
They were featured in the Daily Show. It might have been today, recently. They're getting big.
I mean, and your work, I mean, that just goes to speak to how great your science communication skills are, honestly. Like, it's just awesome that you're doing this in the academia and to the general public as well.
Thanks, Natasha.
Yeah. So I guess, how do you share the science with people? So you share it, how do you share it? Is it just through publications, or how else?
There's so many opportunities. We're really lucky. I mean, we're talking, I'm across the country right now and I'm talking to you guys, which is incredible. So there's like a lot of opportunities that exist today that didn't exist like a decade ago. And I try to take advantage of as many as my schedule allows. So I do stuff like this. This year is the first year that I painfully am not organizing and doing the behind the scenes for Science Cafe, but I know that you guys who are doing an incredible job, and it's a really wonderful opportunity to talk with the public about what's going on in science today. I also do things like That's Life Science blog, which if you'd rather read stuff than listen to it, that's an option. It's all run by graduate students at UMass. So if you're interested in keeping in touch with the community, that's also good for that. I do school visits, and these days I can do it virtually. So I've been able to reach a lot of classrooms through things like Skype a Scientist, which is an awesome program. If anyone who is participating in this science cafe right now is an educator of any kind, we just sign up, and it's totally free, and you'll get to talk to scientists like us about cool research. Yeah, there's like billions of ways. Letters to a pre-scientist, like you get to be a pen pal with a student. So there's all sorts of different ways to spread the science gospel.
I think that's great. And thank you so much for sharing those resources. And like, it's so cool that, you know, anybody could do it. And it's so cool that we're reaching more and more people, and like introducing science to people. Like, I just like the idea of like science being democratized to everybody. So how do you think your PhD helped you in your new steps and your new direction in science communication?
I don't think I would have gotten my job at SciShow without the opportunities that I got in my PhD. So I got a lot of opportunities to spend my time working on Science Cafe, That's Life Science, talking to school classes, working with Girl Scouts, doing all sorts of things that I didn't do before my PhD. But when you're in a PhD program, people just kind of throw opportunities your way. And the more you say yes to, the more that come your way. So I got a lot of practice and a lot of exposure to discussing scientific findings with people who don't work on that specific project. So it's not even just talking to non-scientists, which anyone can be a scientist, but yeah, just talking to anyone about research findings.
Yeah, no, that's pretty cool. And I admire you for that. That's super cool. You definitely have inspired me to get to communicate my science more efficiently. Do you think that, so you do, you're involved in SciShow and a lot of other science communication. Do you think that social media is something that is aiding or hurting this science communication and this communicating science?
So, like you said, democratizing science is like a huge deal. Getting a PhD is not something that while you're doing it is very economically advantageous. As you know, we don't get paid for the amount of hours that we like to put in. And once you hopefully get your PhD, then you can unlock some higher pay grades and stuff, but not everybody can put off that kind of income for the amount of time that it takes to get a PhD. So not everybody can get a PhD. Not everybody wants to get a PhD. That's fine. But reaching more people, getting science to people without having to pay for it, even getting an undergrad education with some kind of science topic like that, it's very expensive these days. So being able to use social media, using YouTube, using TikTok, using podcasts, using all sorts of different media that you can reach more people for free. You can reach people where they're already at. So you don't need to have someone like search for, oh, I want to learn something about science today. Like they could just be scrolling through TikTok and stumble upon something interesting about science. They don't realize that they care about how molecules interact with each other, but they did care that like, oh, hey, like I noticed that my tires are filled with nitrogen when I take them from the dealership. Why isn't it oxygen? Well, the answer has to do with science. So people we trick people into thinking of themselves as scientists. And I think that's really important for people who didn't grow up in a community where it's like all around them. It's not something that we want to keep in certain clusters. We want it to be everywhere for everyone.
Yes, I love that. You don't have to get a PhD to be a scientist. Yes, preach all the time. So last question of the day from me anyway. How have your experiences, how have the experiences and expectations of early career sciences changed across time? Like from previous generations as opposed to now?
Yeah, so I am very fortunate to be in the time that I'm in. I think we're all very fortunate to be in this time, even though sometimes it doesn't feel like it. But it is a lot more of an accepting place for someone like me to be in research. I have generations of women to look up to in science. My advisor is a wonderful female scientist. The first lab that I ever worked in was run by an incredible female scientist. And I've worked along a lot of women in research, and it was never something that people questioned, like, oh, you're a woman in science. Why would you want to be a woman in science? Once I was in it, when I was talking about going into research, there were some questions. And so there's always that stigma still, that being a woman in research is there's still a stigma there, but I have not experienced nearly as much discrimination as people before me. And so you still have like the boys club in research, because in research, just like anything, you've got to make connections, and that's how you progress. And people tell you about their findings because you're friends with them and blah, blah, blah. So people do get shut out of that. And if it's like that for women, it's like that 1,000-fold for any other person that is not historically represented in research. So I'm very fortunate for my experiences, because being a woman is not a minority. It should not be a minority in science. And if you look at any other person's experience, it's still rough, but hopefully getting better.
I like that. Thank you for your candid answer. And I am the same. I'm very fortunate to be in science in this time of day. So we'll get some questions from the audience now. For our first question is, if money was not a limitation, and what's your dream research project?
Oh gosh. I mean, so when I was did I tell a story about when I was in undergrad and how that led to my PhD project?
A little bit. You touched upon it a little bit.
So when I was in undergrad, I was working on attention stuff and in rodents, and we only studied male rodents. Did I talk about that? Yeah. Okay. So my dream project is the project that I went on to do in my PhD, which is like, how many people get to say that, right? That like I was studying the topic that I was interested in, but now I get to study it on my terms and actually compare male and female brains. A dream project is so tough because you have to be in the right place at the right time. So like you need the tools to be like available and created for you to do this project. So like someone had brought up how do we understand autism? I would love to understand autism. I don't think we're there yet. I don't think we have the tools in the background and the fundamental understanding that we need to build. We're currently building it, and then in the future we can have a better understanding of stuff like that. But yeah, it's a mixture of a lot of things, not just money.
Yeah, money is not the biggest limitation at this point. It could be, but not the biggest limitation. It's there for starters. Another question. What was the most surprising thing you've learned in your research?
I mean, I talked about the thing that I find most interesting, which is that like when you're not exhibiting inattention, then like these drugs aren't really changing my rat's behavior, at least. My most exciting finding was probably that I could get the chemistry to work for one of my projects. Honestly, that's something that people don't talk about in research. It's not always like how the finding of like how the brain works that we've discovered this new thing, but it's the finding of like, hey, I could get this method to work in my lab, which is surprisingly difficult. So I mean, it feels very good when you look under a microscope and you see all these pretty colors and you made that happen. So that's, yeah, I guess that's a good finding, that the receptors that I knew were in the brain were there and I could visualize them.
I mean, it's like the finding in oneself that like, yes, you can do this and you have troubleshooting skills to do this. And yeah. And also like, I remember what I was gonna say. Yes, placebo effect is probably the biggest joke like Mother Nature has played on us. It's like, it's crazy. So thank you so much, Emma, for being here with us. And thank you everybody for joining us today. If you have any comments, Emma, or any social media or handles that you would like to share with the public, please go ahead. This has reached the end of our session for today.
Well, my comment is thank you so much for being interested enough to learn about some science that I hope I could share something that you didn't already know. Yeah, if you want to keep up with what's going on SciShow, they're on YouTube, they're on TikTok. They have a podcast called Tangents. They're always making new stuff. So look them up on the internet, emmadauster.com. I post a lot of the outreach stuff that I was talking about, but also the videos that I've helped write. Just reach out to your local scientist and say you appreciate what they do. I guess that my last plug. It's very cool. It's very cool that I get to spend time talking to people about what I do and people actually think it's cool. You know, not every job is like that.
It is super cool. I think the audience really enjoy your talk and what we've learned and what we shared today. So thank you so much, Emma. And our next event will have the same format and will be held on October 21st. And it will be with Leon Gua. So please stay tuned and see you all.
Leon's amazing. Look forward to it.